So I'm very happy to introduce Nicola Fletcher from UCD. So Nicola did her PhD at UCD Veterinary Medicine with Professor Sean Callanan, and then currently is an assistant professor also at UCD in the veterinary medicine department. So I'll hand over the floor to you, Nicola, and we can see your slides, so please go ahead.
Brilliant, thank you very much. Thanks a million for the invitation to speak. I gave this as a very short talk at the recent meeting in Belfast, so this is a slightly longer, more detailed version of it. And thank you, Dallin, for the introduction. So this study really came about as just an idea we had. It was an unfunded project and really a look-see to see whether we had any evidence of SARS-CoV-2 infection in a very interesting fallow deer population living in Dublin, Ireland. Okay, sorry. So it came about because of the multiple reports of SARS-CoV-2 in white-tailed deer in the USA, which can be productively infected with SARS-CoV-2, transmit to other deer, and indeed some evidence now to show you can have reverse zoonosis back to humans. And so, you know, not only ourselves, other groups had asked whether European deer could be potentially infected with SARS-CoV-2. But initial reports indicated that there was no evidence of SARS-CoV-2 infection in multiple deer species in the UK, Austrian and German deer populations up to 2021. But from 2020 up to 2022, we evaluated this population of fallow deer for SARS-CoV-2 infection. So the animals are located in the Phoenix Park, Dublin. It's Europe's largest city park, 1,750 acres in Dublin city. It is just north of the city centre, so you can actually see in the top left image the train station to the left-hand side. And then that circular building is the Central Criminal Court, so it really is quite central. And then you can see the park with a huge road running up through it, leading to the north side of the city. And the animals, there are about 650 resident fallow deer, so dama, located in the park. They were first brought to the park in 1662 and have been there ever since. They're managed by the Office of Public Works, and there are many behavioural and other studies that have been done and are being done on an ongoing basis on these animals, so they are very well studied. About 80 to 85% of the population is individually recognisable, so they have coloured ear tags with an individual number on them. And there are managed culls yearly, so approximately 10% of the population is culled every year, and it is done by trained marksmen, but it's in a random cull. That's the intention, is to maintain the natural sort of population of males and females and age profiles, etc. So the animals, this is just a simple diagram, but there are many subherds within the park. And on one side of the park is where the females tend to be located, and the males are mainly in the other side of the park, but there are smaller subherds. And then the males cross the road to mate with the females during the rutting season. And most of the behavioural work has been led by Dr. Simone Cuti, who's based in UCD as well. And it was really through a collaboration with him that we were able to do this study at all, and his behavioural knowledge of the animals. So the animals are studied throughout the year quite closely. The fawns are caught in the spring, and they are tagged, weighed, and measured. And then literally the amount of time it takes for the fawns to run away from the handlers after they are released is noted, and many other behavioral observations. And one of the really interesting studies from our point of view was carried out by Laura Griffin as part of her PhD, where she studied the animals' interactions with humans. So the animals, they are basically quite tame, most of them, and they will approach humans and take food from them and allow themselves to be petted, etc. So you can see Laura taking her observations there, and a child going close to those 3 deer. And, you know, I'm not exaggerating when I say they have very close contact with humans. You can see there a chap giving a deer a drink of Coke from his bottle. So just to talk a little bit more about the begging behavior of these animals, from Laura's study, she found that in general, males are a lot more likely to take food and beg from humans than females. And the animals they all have a begging profile. So they've been assigned into begging categories, from frequent beggars to occasional beggars to avoiders. And then some of the animals, I mentioned that 85% of the animals approximately are ear tagged. So the untagged animals and the young animals would be unranked. So there have been many initiatives by Simone Cutie's group, including Laura, and also the Office of Public Works, who manage the deer, to try and restrict the frequency of people approaching them and feeding them. It is, I suppose, of some success, but you know, they have a lot of interaction with humans, particularly during the COVID pandemic in 2020, where there was a large increase in the number of people entering the park and interacting with the animals, because of the lockdowns we had, where they were only allowed to travel 2 kilometers or 5 kilometers from their home at different times. So they used the park for recreation if they lived within the vicinity of the park. So in terms of our study, we screened the fallow deer for SARS-CoV-2. In November 2020, we had access to 53 stored sera from 2 cull dates in November. And then from 2021 to 2022, we attended 3 culls and sampled the animals ourselves. So the first one was the 2nd of November 21, the 25th of November 21, and then the 16th of February 2022. So about 20 to 25 animals culled each time. And our sampling strategy was initially not totally looking for SARS-CoV-2. We were also looking for hepatitis E virus. So we took, following culling, about an hour after culling, we took blood, and isolated sera from that, and then we took retropharyngeal lymph nodes, tonsils, nasopharyngeal mucosa, brainstem, and then cecal content, liver, small and large intestine. And the samples in red are the ones we initially screened for SARS-CoV-2, based on the studies that had already been published from white-tailed deer. The initial test we used was the GenScript CPAS SARS-CoV-2 neutralization antibody kit for the sera, and we did qRT-PCR for SARS-CoV-2 and E gene. And then the plan was that if any of the samples were PCR positive, we would test the remaining tissues in black, we'd attempt culture of the virus, and we'd do pseudovirus neutralization assays if we found any seropositive animals. So here are the data from the GenScript CPAS SARS-CoV-2 neutralization antibody ELISA. In November 2020, you can see that all of the sera were below the limit that would be considered positive, which is 30% in this assay, and that's indicated by the dotted line. 2021, you could see they were starting to rise a little bit, and we did have one animal marked in red, which was actually right on the cutoff, so it was 30% neutralization, so that would actually be considered seropositive. And then in February 2022, we had a number of animals that were over the limit to be considered seropositive. It was actually 57% of all of the animals we screened in February 2022. And at all time points, retropharyngeal lymph nodes, nasopharyngeal mucosa, tonsils, and cecal content were all SARS-CoV-2 PCR negative. And so just looking at the seropositive animals in a little bit more detail, and we linked this back to the begging data that Laura Griffin had generated. So in all of the time points, the begging ranks, the distribution of them were quite similar. In February 2022, you can see that the seropositive animals were all either consistent or occasional beggars. And then we did also sample some untagged animals, which would be considered unranked. So we don't know their begging status. And then on the bottom, just briefly to say that the begging ranks and categories from the sampled populations were representative of the entire population of animals in the Phoenix Park. And I haven't shown you the data here, but 77% of the seropositive animals were male. And this sort of would fit with the idea that male animals are more likely to approach and take food from humans. So we took the three sample dates we had: November 2020, November 2021, and February 2022. And we looked at what variants were circulating in humans during those sampling periods. So in November 2020, we had mainly B1 and B1177 with alpha and zeta. We had just one sample of zeta towards the end of the month. In November 2021, it was pretty much exclusively delta that was circulating in the human population. And then in February 2022, Omicron emerged with BA1 at the beginning of the month and a few samples towards the end, and then Omicron BA2 also there with BA3 towards the end of the month. And so we generated some pseudoviruses based on some of the different circulating variants that were present in the human population when the animals were sampled. So we generated SARS-CoV-2 pseudovirus with alpha, delta, BA1, and BA2 spike proteins on them. And we saw that, so the pink sample is the sample from 2021 that was right on the cutoff. It was 30% neutralization in the CPAS assay. And we saw that it didn't really neutralize the pseudoviruses, with the exception of BA2, which it did significantly neutralize. However, the 4 samples that are shown in green are 4 of the highest titer viruses from, sera from the 2022 cull, and all of them neutralized all of the SARS-CoV-2 pseudoviruses that we looked at. We didn't see any neutralization with any of these sera of vesicular stomatitis pseudovirus, which is an unrelated pseudovirus, just as a control. And then we looked at some infectious SARS-CoV-2 and the ability of the sera to neutralize those. And this was quite a bit more variable, but in general, I think, so Italy INM1 is representative of the ancestral virus, so the Wuhan virus. And then we looked at a clinical isolate of BA1 taken from an Irish patient. And the sera, some of the sera from 2022 were able to neutralize the ancestral virus, but all of them neutralized BA1. So to look at this in a little bit more detail, we generated fallow deer precision cut lung slices, and we also generated tracheal explants, which we cultured as an air-liquid interface, and we challenged them ex vivo with either the ancestral virus or the clinical isolate of Omicron BA1. So the deer lung tissue was taken back to the lab immediately after culling. It was subsequently shown to be seronegative, or these animals were seronegative for SARS-CoV-2. We perfused them with agarose and then put them into an agarose mold, and we cut precision cut lung slices of 350 microns in thickness. And we cultured them in lung slice media we used an excess of media because we wanted to make sure that they remained perfused. So you can see the lung slice or the precision cut lung slices in the bottom right image just before they were infected with SARS-CoV-2. And so these are just to show you the tracheal explants. Infecting them with the ancestral virus, we saw minimal infection of ciliated epithelium in just one of the 2 animals that we infected, with no infection of the IgG controls. But in contrast, when we infected the precision cut lung slices from the fallow deer with the ancestral virus, we saw no infection or no infected cells whatsoever. But with BA1, we saw infected cells within the alveoli, which are morphologically consistent with type 2 pneumocytes, which are similar cells to those that would be infected in human lungs. We had these evaluated blind by a human histopathologist with experience looking at human COVID lungs, and also by a veterinary pathologist. So in contrast with Stefan's work, I suppose these tissues will not have any inflammatory infiltration because they were infected ex vivo. These are shown at 72 hours post-infection, which is the only time point we looked at, just to we really did a best guess here. We weren't sure what we would find, but we did see that, yes, Omicron BA1 infected them, despite the titer of this virus being slightly lower than the ancestral virus that we used. So to conclude, we have seen that fallow deer in a city park in Dublin are seropositive for SARS-CoV-2, and that the seropositive deer that we had a begging category for were all consistent or occasional beggars taking food from humans. The SARS-CoV-2 seropositive sera neutralized a range of SARS-CoV-2 pseudoviruses and infectious virus. Omicron BA1, but not the ancestral virus, infected the fallow deer lung tissue ex vivo. So to our knowledge, this is the first description of SARS-CoV-2 seropositive deer in Europe, and the first non-white-tailed deer in which SARS-CoV-2 antibodies have been detected. It's up on bioRxiv if anyone would like to look in more detail, and was profiled by Science this week. So there's a nice article about it there. And I'd really like to thank my research group for, taking time out of their projects to spend quite a bit of time on this project, particularly Dr. Kevin Purvis, shown in the bottom left image, standing on the left. This whole project was his idea, and I really can't thank him enough. And to Hannah Brown, Ruth Haverty, Habib Jimmy, and Chris Evans for their help. And also to Simone Cutie and his group, particularly Laura Griffin, for their help with this project. Thank you very much.